Your Oncogens, a presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. There are two cancer manifestations, genetic cancer and clinical cancer. Both maintained homeostasis, and homeostasis is a self-regulating complex system by which processes and organism maintains stability and maximal optimality. So here you have the two representation of cancer, the genetic cancer, which is homeostasis of processes, and clinical cancer, which is homeostasis of the upper presentation of cancer. Now, both cancers are studied and represented by this milieu anterior, which describes all what is found in the genetic cancer or clinical cancer. So we are talking about proto-oncogene and oncogene. Proto-oncogene is a normal gene that could become an oncogene due to mutations or increased expression. Oncogene is a gene that has the potential to cause cancer. So here you have, they are defined in the genetic cancer but studied in this milieu anterior. So let's investigate two homeostasis states. State 1, we are interested in the proto-oncogene, and state 2, we are interested in oncogene. But we remember that in homeostasis, many processes interact or affect our proto-oncogene or oncogene. Although we treat them as separate entities, but we know that they are processes which they represent processes. So in state 1, we see the proto-oncogene, state 2, we see an oncogene, and we ask, is it possible that state 2 becomes in state 1, which means oncogenes become proto-oncogene, and we know that it's impossible. There are no observations where oncogene become proto-oncogene. This example illustrates the limit of oncogene theory. Proto-oncogene progression cannot be reversed, and oncogene theory does not suggest any oncogene therapy which might solve this problem. Recently, I mentioned a study that provides a solution to this problem, and it was published by Rubin and Vogt, an avian leukosis virus associated with stocks of Rau sarcoma virus, and I discussed this also in a previous presentation, viral interference with SARC oncogene. They describe interference between two viruses, Rau sarcoma, RSV, and Rau's associated virus, RAV. RAV does not produce any cytological alternation, and RAV is detected since blocking RSV. When viewed in the electron microscope, RAV is morphologically indistinguishable from RSV. So what does it mean? That both viruses, RSV and RAV, carry the same SARC oncogene. apparently Sark in Rav is inactive, and this important observation will be discussed in future presentations. Not all Sark oncogenes progress to genetic cancer. More to come. Enjoy.